Previously, you learnt about making direct wet smears with saline or iodine. Wet mounts can also be prepared from concentrated fecal specimens. Concentration techniques increase the chances of detection of cysts and eggs in the fecal specimen. Therefore, concentration techniques are useful when cysts and ova are too low to be detected using a direct wet smear. It is usually recommended that stool samples are concentrated as a routine measure. These concentration techniques are designed to separate protozoan cysts, helminth eggs, and larvae from fecal debris by centrifugation and/or differences in specific gravity. Though protozoan cysts and worm eggs and larvae can be concentrated, trophozoites cannot be concentrated because they are usually destroyed during the procedure. There are two concentration techniques. One is the flotation technique. Here, we separate cysts and eggs from bulk of the fecal matter by floating parasite stages on the top, using a solution of high specific gravity like saturated sodium chloride or zinc sulfate. The second concentration technique is called a sedimentation technique. In the sedimentation procedure, we separate cysts and eggs from bulk of the fecal matter by sedimenting parasite stages at the bottom using centrifugation or a solution of low specific gravity. Solutions used for the sedimentation procedure are formalin ether or ethyl acetate. We will look at the sodium chloride flotation technique in this presentation. These are the learning outcomes of this exercise. In the flotation technique, parasites in fecal samples are floated in a solution of high specific gravity. This procedure will permit the separation of protozoan cysts, coccidian oocysts, microsporidian spores, and certain helminth eggs and larvae. Flotation techniques give a cleaner preparation than does the sedimentation procedure. And it is a simple method that does not need any sophisticated instruments. The material required for the procedure are shown here. To start, take 2 ml of saturated sodium chloride solution and add it to a beaker. Then take about 1 gram of feces and add it to the sodium chloride solution in the beaker. You must mix the feces with the sodium chloride solution thoroughly to make an even suspension. Take a 15 ml tube and keep a funnel on it. Then take a wet strainer and filter the suspension through the strainer into the test tube. Now add some more saturated sodium chloride solution until the solution reaches almost to the top. Add the final drops using a pipette to give a convex solution surface over the brim. You must take care not to overspill. Your preparation should have a convex surface like this when observed from the side. Take a cover slip and carefully place it over the top of the solution. You must do this very carefully, making sure there are no air bubbles trapped under the cover slip. Now you can leave the test tube undisturbed with the cover slip in contact with the solution for about 30 to 45 minutes. With time, fecal debris will be seen at the bottom of the tube, and the parasite stages will go up to the surface film. After 30 to 45 minutes, gently raise the cover slip with a direct upward lift. You must take care not to spill any liquid. Place the cover slip on a clean slide with the wet side of the cover slip placed down. Now the preparation is ready for microscopic observation. You must examine under the microscope the same way as described for the direct wet smear. Systematically scan the cover slip area first with the 10 times objective. Use the 40 times objective if something suspicious is seen for a more detailed study. You will be able to see any eggs present easily in a very clear background. If necessary, you can add iodine to enhance morphological detail. You must examine this concentrate within 15 minutes. If left for too long, the high osmotic pressure of the solution will distort eggs and cysts, making it impossible to identify. Some limitations of the sodium chloride flotation procedure are shown on the screen.